Hello, students. Welcome back as we continue our discussion from yesterday about DNA. 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 Let me hear you. DNA. 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 All right, very good. So what would you rather say, DNA or deoxyribonucleic acid? Say it out loud. All right, enough fun here for today. Let's get started right away. We've got a number of things to discuss regarding our DNA today. So let us proceed to where we left off yesterday. Yesterday, if you recall, we were learning about nitrogen bases of DNA. And let's see. Ooh, let me block them here for a moment. Let's see. Little quiz time. I'm going to give you a letter. I want you to yell out what you think the correct nitrogen base pair is. Here we go. A. T. G. C. All right, not bad. Quiz time. What nitrogen base pair is always paired with adenine? Thymine, very good. Which nitrogen base pair is always paired with guanine? Very good, cytosine. And remember the little memory aid from yesterday? What happens to our turtle friend when it gets cold outside? A turtle gets cold. That's right. All right, so today let's pick it up and talk about, not that, but let's talk about the replication of DNA. The replication of DNA. Raise your hand if you can tell me what replication means. All right, if you said something like the duplication and division of it, fantastic. That's what replication means. To reproduce something, replicate, reproduce, the replication of DNA. And as you may recall from some of the videos earlier in the week, it is super duper important for this to happen. The replication of DNA is so vital because the DNA and this sequencing of nitrogen base pairs is what carries the information that determines who you are from a biological point of view, at least. It is what determines the characteristics of a living organism. And so if you get a boo-boo on your skin and your skin cells want to produce more skin cells to heal the boo-boo, you got to have skin cells with the skin cell DNA in them to make more skin cells. I mean, imagine if your skin cells had the wrong information and started making bone cells, and then you'd have bone growing out of your skin. Or hair cells. You get a hairy boo-boo. Every time you got a boo-boo, you'd get hairier and hairier. I mean, think about the consequences here if we didn't have the appropriate replication of DNA. So how does it work? Well, let me show you. We're going to try and narrow it down to 3 simple steps. The first thing that happens in the replication of DNA is that the DNA itself unzips. It zips down. Now, I don't have a zipper on my jacket. I've got buttons. But imagine you had a zipper on. You can zip it down, zip it up, zip it down, zip it up. In fact, if any of you have a zipper jacket on right now, do that a few times and show everybody else in the class. Hey, you can zip it down and zip it up. If your zipper's on your pants, Leave the zippers alone, however. Please don't demonstrate pants zippers. Okay? So, duplication of DNA, the DNA unzips. I want you to think about, in this example, in this analogy, think about a zipper as if it were a strand of DNA. And the little teeth on either side of the zipper represent the different nitrogen base pairs. So the first thing that has to happen is that those nitrogen base pairs come apart. You're like disassembling the puzzle pieces that go together. You are unzipping the DNA. But it can't stop there, because the second step is that the free nitrogen bases then pair up. Well, what does that mean? Well, first of all, what is a free nitrogen base pair? Let me just go up. Don't worry. I'll get back to that slide in a moment so you can finish copying it. The nitrogen bases that are normally together in the zipper would be paired. When you unpair them by unzipping them, they are now both free. Free means by itself. So the free nitrogen bases, free means by itself. They, by nature, want to pair up with something that fits it. It doesn't like to be alone. So these nitrogen bases that are now free, they're separated from their original pair. And by the way, I hope you see the play on words here. Pair, P-A-I-R, is not the same as P-E-A-R, but they're pronounced the same way because they are homophones. Anyone ever watch VeggieTales? There's a great song about homophones in one of those episodes. But anyway, the free nitrogen bases find another free nitrogen base that is its perfect match. It's kind of like the old movie with Tom Cruise in it called Jerry Maguire. There's one scene in that movie where Renee Zellweger and Tom Cruise are in an elevator and they realize they were meant for one another. And he says to her, "You complete me." It's like two free nitrogen bases coming together because they complete one another. It's a love story every time your cell divides. Now, it can't just stop there either, because they have to pair up in the correct fashion in order for there to be two new DNA molecules to form. Now, I have a picture here in the background that kind of illustrates it fairly well. We start out with our original, let's see if I can point to it this way. We start with our original strand of DNA, which then becomes unzipped at this point here. And each half of our unzipped DNA strand, these free nitrogen bases find other free nitrogen bases floating around in the nucleus of the cell to pair up with, so that they form two new strands of DNA molecules, which are exact mirror images of the original DNA. So what we start off with as one strand ends up as two strands of DNA. Let's go to a close-up view to get a better idea of how this happens. If you look at our strand of DNA, and we'll look at the letters this time, you can see how each of the letters are paired together. It's as if we unzip the strand of DNA. And as we do that, All of these free nitrogen bases are now sad because they would prefer to be together. But once they separate, they don't come together with the same nitrogen base pair. They look for another matching nitrogen base pair floating around in the nucleus so that they can both match up with other free nitrogens to create the two identical but mirror images of the original strand of DNA. Only through this process can you get identical copies of the original. It's actually a fabulously efficient method that takes place billions of times in your body every day. It's unbelievable how efficient this is. Are there ever mistakes? Yes, and stay tuned for that. But for now, let's dive in a little bit deeper into what is going on and why. And here it is. We have talked about chromosomes before in the past, and now's the time to unpack the chromosome a little bit more. Last week, when we learned about cellular division and mitosis, we learned about how the chromosomes duplicate and divide in the nucleus during mitosis. Well, what exactly are these chromosomes? Well, check out this picture. This is a great picture that illustrates what these are and how it all relates to this week's topic. So let's begin with the cell. You know how small a cell is to begin with. We need a microscope to see inside, but we've seen cells and we've seen the nucleus inside of a cell. And we know that inside the nucleus of a cell, we've got chromosomes. And we talked about the V-shaped chromosomes, and here we actually have a pair of chromosomes or a chromatid together. It's a topic from last week, if you recall. But let's just say here we have a chromosome here, this purple structure. Well, these chromosomes are made up of coiled up DNA. Remember yesterday how we talked about the double helix shape of DNA and the efficiency of squeezing information all together. Well, there's a lot of information in DNA, and you know how small a cell is. You've got to efficiently pack away all of that information into a tiny, tiny, tiny little area. So the way it does that is that it coils up on itself. But if you take the DNA and you unravel it or stretch it out, which can be done, you can unravel the chromosomes rather. It stretches into these strands of DNA. The DNA is made out of these nitrogen base pairs that only go together in one way. And different sections of the DNA are what we call genes. And we'll be getting into that a lot more later on. But genes, G-E-N-E, not G-E-A-N, genes are little segments of DNA strands that determine specific things. Like there's a gene that determines my eye color. There's a gene that determines my hair color. There's a gene that determines my leg length, and there's a gene that fits my leg length. It's I think I take 32 long, and well, I used to wear 32 waist and 32 length, but the older I get, the bigger that first number gets. I've been up to 38. All right, enough about that. All right, I'm not talking about Jean, talking about Gene, the genes that determine your characteristics or your physical traits. Again, we're going to pack that a lot more in the not too distant future. But chromosomes. Let's write this down. Chromosomes are rod-shaped structures. And these rod-shaped structures are found in the nucleus of a cell where genes are found. And again, we're talking about Genes, genes. The genes are on the chromosomes, and the chromosomes are packed away in the nucleus, and chromosomes are made out of strands of DNA. So chromosomes, rod-shaped structure in the nucleus where genes are found. Now, there is a theory called the chromosome theory. And there's a lot of information in the chromosome theory, but the basic point of the chromosome theory is that the genes, or those little snippets of DNA that determine the characteristics or traits of an organism, are carried from parents to offspring. And I have two pictures here. One of these pictures represent the mama chromosomes, and one of them represent the papa chromosome. Now, by looking at them, you will notice that both of them have 23 pairs of chromosomes. But could anyone look at these two chromosomes and determine the gender of the chromosomes? Anyone wish to take a guess? Well, I'll give you the answer here. In the first case, on the left side, and these are different types of images. These are actual photographs of chromosomes on the left and just a diagram on the right. But they basically look the same under magnification. But you'll notice that, the 23rd chromosome on the left here is actually two X chromosomes. There are two X chromosomes. This is the chromosome for the female gender. Girls, women, mommies have only X chromosomes, whereas boys, men, and daddies have both an X and a Y chromosome. And as we will discover through our discussion of genetics, when a mommy and a daddy really love each other very much and they get together and have a baby, half the chromosomes from the mother, half the chromosomes from the father get together, and the genes or traits found in the DNA of the chromosomes, half of them are from the mom, half of them are from the dad, and the resulting offspring will be a combination of both parents. Now, here's the deal. The mother's 23rd chromosome can only supply an X chromosome, a female chromosome. It is the male, actually, that has both a female and a male chromosome. So if the daddy's X chromosome gets together with the mommy's X chromosome, then they're going to have a gender reveal party with probably the color pink coming out of a pumpkin or a firework or something. They're going to have a little girl. If the daddy's Y chromosome joins together with a mommy's X chromosome, then the resulting offspring is going to be a boy or a male, and they're going to have a different gender reveal party. By the way, when I was a kid, the gender reveal parties were called birthdays. Parents didn't know until a child was born what gender it was going to be, and that was the gender reveal party. But hey, times have changed. So the times, they continue to change. The times, they are a-changin'. All right, now let's get back to the chromosome theory here, which states this. I mentioned it earlier, but now's your time to write it down. The chromosome theory states that genes, and remember what genes are, they're little segments of DNA found on chromosomes that determine a certain characteristic or trait. It is the genes that are carried from parent to offspring, and they are found on chromosomes. And again, chromosomes are rod-shaped structures that have coiled up DNA. They're made up of coiled up DNA to take up less space in the nucleus of a cell. So the chromosome theory states that genes are carried from parent to offspring. Now, in humans, as an example, human beings have 23 pairs of chromosomes. Now, the word pair, hopefully you know what the word pair means. It means two of something, which is why I showed those two pairs earlier in one of the slides. But if humans have 23 pairs of chromosomes, then what would the total number be? Hopefully you said 46. 46 is 23 times 2. Fruit flies, on the other hand, fruit flies, on the other hand, have 4 pairs. So what would the total number be of fruit flies? 8. 4 times 2 is 8. So different species have different numbers of chromosomes, and it's not just the complexity of an organism that determines the number of chromosomes. There are actually organisms that have more than 23 pairs of chromosomes. Don't ask me to name one. I can't think of one off the top of my head. Maybe you can see one. Perhaps not, because there's less up there to see these days, which was determined by one of my genes that I inherited from my parents. But let's continue on with chromosomes and DNA, because there's a very important topic we have to discuss, which is going to get to our lab activity tomorrow. And it has to do with this word meiosis. Now be careful. Last week we learned about mitosis. Meiosis is not the same thing as mitosis. Does anyone remember from last week what mitosis was? Mitosis is the duplication and division of a cell's nucleus. Meiosis, however, is simply the production of sex cells. Now, before you giggle too much, ooh, sex cells. No. Any organism on planet Earth that undergoes what's called sexual reproduction, meaning there's a mommy and a daddy of whatever species we're talking about, undergoes meiosis, because they have to produce sex cells so that each parent can contribute half of its chromosomes to the offspring. So let's get into a little discussion about sex cells and more generally meiosis. This is super duper important. During the production of sex cells, which are in key ways very different from normal cells, in that they have only half the normal number of chromosomes. Think about it. Think about it for a moment. We've got organism A and organism B. If organism A and organism B get together and produce offspring, before that can happen, each organism has to divide its chromosomes in half. Otherwise, when their sex cells would join together, the offspring would have 2 times the number of the normal number of chromosomes, and bad things would happen if that were the case. So sex cells have to be produced in order for each parent to have only half the normal number of chromosomes. And this happens through 2 cellular divisions. Now, this is different from last week's topic of mitosis. So it's going to start off looking the same, but there's one big important difference when we talk about meiosis. So to illustrate this, we're going to draw a picture. Actually, we're going to draw a couple of pictures on the bottom of your notes page. So let's begin with the one that's a little bit simpler, which is the male meiosis. During male meiosis, we start off with a regular cell. Now, this is going to become a sex cell, but we're going to look at it in terms of chromosomes here. And I have drawn our chromosomes as different length lines of different length. We're not going to draw millions and millions of chromosomes here. As you can see, I've just drawn 4. 2 long ones, 2 short ones. In the first part of meiosis, we have something similar to mitosis. The chromosomes found inside the nucleus of the cell duplicate and divide. So notice what happens. We start off with a total of 4 chromosomes here. How many are there now? Twice that number, which is 8. Okay? So we've got double the normal number of chromosomes. But this can't stop here. We can't use this as a sex cell, because if the male and the female both had double the number of chromosomes, well, the offspring would have quadruple the number of chromosomes, and that would just not work. So the first thing that takes place after the duplication and division of chromosomes, which is kind of like interphase of mitosis, is we eventually have our first cellular division. And in our first cellular division, notice the number of chromosomes that we currently possess. We started off with 4. Excuse me. We duplicated and divided. And now we're back to the normal number of chromosomes again. Each of these cells has 4 chromosomes. Now, if this were normal mitosis, this is where it would stop. But this is not mitosis, this is meiosis. And so, remember, in meiosis, we have to produce sex cells with half the number of chromosomes. So what happens is a second cellular division takes place. And make sure you're labeling this diagram in your notes with the first division and the second division as well. So after the second division during male meiosis, we get the production from one original cell, we get the production of 4 sex cells that probably have an appearance that looks similar to you. They look like little tadpoles. And that's what they look like. They have tails. They are produced with tails that enable them to swim. And those cells are called sperm cells. Now, every living organism that undergoes sexual reproduction, the male of the species produce 4 sperm cells from every original sex cell. That sperm cell can take on various appearances, say like flowers. Does anybody know what the sperm cells are with flowering plants? Anybody? Some of you are allergic to it in the spring. It's called pollen. Pollen are the sperm cells of flowering plants. So sperm cells can take on different appearances based on the species of organism. So here we have male meiosis, and I'm going to show you a picture now of female meiosis and give you some time to copy this down. The steps are the same with female meiosis. We begin with a first sex cell that undergoes the duplication and division of the chromosomes, undergoes cellular division, and then it undergoes a second cellular division. But look at how this differs. In female meiosis, there is only one viable egg cell which is produced, and three what are called polar bodies. You see, females take only the best. They are more selective. They look for only the best. And so females will only take the best sex cell, which results in one egg cell, and all of the other ones that are substandard disintegrate and are eliminated out of the body. They are called the three polar bodies. So here we have male and female meiosis taking place over two divisions in order that the final sex cells produced have half the number of chromosomes. Very, very important points in meiosis. And so we're going to stop it here for now, and we'll pick it up tomorrow for our lab activity, where we perhaps do some interesting things with meiosis. So until then, I am going to say bye-bye.